and as a result, you'll need to do different methodologies to do so. So I'm sure many of you are familiar with MD. And so that is typically at the atomistic scale, and you can get into nanosecond, microsecond regimes with how long you're able to simulate. And so therefore the processes you can look at. And so when you're trying to do whole cell simulations or processes that happen on cellular timescales, where you're looking at more second to hour regime, you need to use different methods. One example of that being the RDME, but you need to use more numerical methods that are able to simulate at those timescales and deal with the species copy numbers that you're dealing with when, let's say, looking at a gene regulatory network. So I go to another slide. It's another example of kind of what you just saw. It's just the idea of that you need different methodologies, so dynamics, the reaction diffusion master equation, CME, chemical master equation, ordinary differential equations, and partial differential equations, depending on the type of process you're looking at. And so in our group, and when looking at whole cellular processes, the most, I shouldn't say most important, but a very important part of it is obviously looking at gene expression due to the fact that is how all functions are essentially made and or coded for in a cell. And so when trying to do gene expression and metabolic networks, you need to use these regimes at the top of this figure, which is the chemical master equation and the reaction diffusion master equation. And so here's a list of some different cell simulation techniques. But as I've kind of already touched on, we're going to be focusing more on the probability-based methods, which would be the reaction diffusion master equation and the chemical master equation. And so here are some examples of some simulations that we've done looking at E. coli cells and some gene regulatory networks. There are some tutorials that will kind of allow you to do some related simulation types of this, or at least analyze some data related to these type of simulations where you're looking at a genetic, the lac genetic regulatory network in E. coli. So you have gene expression events being controlled by some inducer, which allows for the continuing of the gene expression events or the stopping of them to allow some process to happen in the cell. And so if I keep going forward, we're using these probabilistic methods. And again, some of this is going to be a review because Zan touched on it, but just to kind of reiterate and emphasize some of the important notes is that we talk about using these probabilistic methods. And so why are we using these probabilistic methods? And that's due to the stochasticity in biology. So I believe, again, Zan talked about this experiment by Elowitz et al. In Science. And so what they were able to observe is the stochasticity or randomness in genes or in biological life. And so the idea is that the cellular events appear to be random or stochastic in nature. And one way you can kind of see this is if you think you have a colony of cells with all of the same genome, so everything that they're encoded for is exactly the same and they're under identical conditions, or you can assume identical or homogeneous conditions based on the fact they're within the same media. You're seeing different expression events or different numbers of proteins, RNA being formed, and that's due to the stochastic nature. And so what this experiment did is they put two reporters on opposite sides of the circular genome at equal points. And so if biology was deterministic, what you would expect to see is that both of these genes would be copied at the same time, and therefore their messengers would be produced at the same time. And thus, you would have both green and red fluorescent proteins being expressed. And so you would get some combination of them presenting in some yellow color. And so by doing this, they were able to directly measure it. And so what they find is contrary to what you would think if things were deterministic, is that you get a distribution of colors from this expression platform. And so what that's telling them is that there is some sort of randomness at which these the red and the green fluorescent protein were being expressed. And I have here in the last kind of bullet point is it's important at low copy numbers. So your noise or your variance here is directly proportional to your copy number. So, or inversely proportional, I'm sorry, to your copy number. So if you have lots of particles, it's going to not be as impactful. And so where you kind of see this is if you think of doing gene expression models where you have a single gene, max 4 genes potentially, depending on gene replication states. And then you have something like metabolism where you have, let's say, ATP molecules, which are in large quantities, typically millimolar concentrations. So although there is still some stochastic nature to those reactions with the ATP molecules, it's not as, or the noise that you're seeing is not as significant due to the fact that you have so many more particles. And so that can kind of be seen here in this simple kind of example where you just have a generation of species A and the decay of species A when simulating it stochastically, and you have a small particle copy number, so no more than 40. You can see the variation, and then you can also see the distribution of the noise in the data. When you have a large copy number of A, so thousands of particles, you can tell, and again, your distribution or your width of the data is represented by the blue here. You're seeing that it's much more similar to what you would consider a deterministic experiment. And that's due to the fact that your fluctuations are typically 1 or 2 particles, roughly, depending on your rates in the system. And so if you have a system where you have thousands of particles, those fluctuations are not going to make a significant impact. And so kind of going along there, touch a little bit about the concepts that Zan touched upon earlier to just reiterate them. And so we have a program that we use in our group, in the Luthi-Scholten group, and I'm showing here from our the group website, where you can get some more information about the program. And then there's also some information to download it and get some tutorials slash the user guide for it. What I will note is that this is LM 2.3, which is the version you'll be using today. It's pretty similar to LM 2.4. However, there are some new modifications that we've added. 2.4 is not currently out yet, but we are currently working to submit a manuscript that Zan touched upon. And then once that is submitted, we will work on submitting something else that would be a more technical paper talking about some of the modifications we made to Lattice Microbes. But the big thing about it is it's just a suite of solvers that are able to do well-stirred and spatial simulations. It uses GPU, so it can be quite fast depending on the system and how things are optimized. And it can be run through the command line because it's a C program, but it has a Python wrapper, which I'm kind of showing here in this little diagram, which is referred to as PyLM, and then this STD LM PyLM, which has to do with the output, more of the processing side. That allows you to use Python, the Python language, to interact with the program. And that's how you'll be interacting with it today through the Amazon web servers. And so that's Lattice Microbes. Very kind of quick overview. Again, the website, and I can send that link around to get more information on it. But today we'd like to stress more doing the tutorial, so just giving a little brief overview. And so here's a kind of schematic or diagram of building models using PyLM, so using the Python interface of Lattice Microbes. And so it's pretty much, you need to determine the type of simulation you're going to do. So if you want it to be spatial, if it's an RDME, then you're going to need to define regions. And so what all of these things are just different steps of it. And so the idea being, if it's a spatial simulation, you're going to need to have, sorry, there's a message in the chat. Let me move that. Oh, thank you.
I just dropped the link to this.
Thank you. No, I appreciate that. And so the idea is just kind of following this diagram. So if you're trying to do a spatial simulation, you need to define those spatial regions. And an example of this is, let's say you're doing a eukaryotic cell. If you have something that's in the nucleus, it might be able to diffuse outside of the nucleus through the nuclear pores. And so if that's the case, you need to define those spatial regions and also the events that can happen between the spatial regions. Another example of something where spatial regions would come into place is if you have membrane proteins, you could have them diffuse to the membrane and then be inserted through the sec-Y system if you're thinking of bacterial species. So it's all about the information you kind of have about the system, and then you can put it in some capacity. And Zan touched upon the idea of adding in geometries. This will be a part of the tutorial, but the idea is you can take experimental data directly and use it to optimize or build into these different components, whether it be taking cryo-EM data to figure out their structure, kinetic data to determine reaction rates, and then also someone in this species name, species identification is using a lot of times super resolution imaging data where you're able to quantify species in the system. And so if I continue on, I have at the bottom simulate using the chemical master equation. So to just quickly go over that again. So the chemical master equation is a probabilistic equation that allows you to sample different states of a trajectory. And so the way we use it is what you can do is assume state S is a vector that describes the copy numbers within your species or within your reaction. So how many RNA, how many proteins you might have. And then what you can do is think of the reaction think of the chemical master equation as a way to move between states. So what you do is you describe your system as transitions between different states. And so this first term this term here that's boxed is transitions out of your current state. So if your current state is S, it's transitioning to some other state. And so S-1 could be, you could have a degradation event occur, for example, and then S+1 or +2, you could have a transcription or translation event occurring where a new species is being generated. Then the other part or term in it is transitions to your current state. And so it's essentially the opposite, but the idea is you're moving from an S1 or S-1 or an S+1 state to your state S. And so there are always some sort of kinetic rate that allows you to transition between these states. And this alpha term is called the propensity, and that is related to the probability of transitioning between the states. And so here's an example of a small stochastic gene expression network done through the CME. And so here's your basic reaction scheme where you have some gene that is able to transcribe messenger M at some rate, and then that messenger can decay. So a very simple schematic. What I have here is the ODE version of it. So if you were trying to solve it using an ODE, you would set up the right-handed side using this, and you could be able to evolve trajectories. And so in the chemical master equation, it's a little bit more complicated, but again, you have your different transitions, whether that be a gene being made, a gene being decayed, or no event happening. That allows you to describe all of the events in the system. And so continuing forward is what you can then do is use the chemical master equation to sample. Again, as Zan said, you're not solving the chemical master equation. You're sampling it using the Gillespie algorithm to generate trajectories. And so what's super interesting is if I keep going, kind of go towards some of the later runs, is that running with the same exact initial conditions, you will get the colored lines you're seeing. So the green, red, and blue lines where you're seeing different trajectories evolve throughout time using the exact same kinetic scheme, whereas the black line would be what you would get from an ODE or a deterministic method where your input is directly going to correlate to the answer you get, and that will not change. And so again, if you do multiple replicates, you can actually get a distribution of results, which is shown in the green, and the blue is your average, because the more simulations you run with the chemical master equation, you will eventually get out the deterministic result. And why some of this is so important is with techniques like super resolution imaging, where you can now get single cell, single molecule resolution, you can now use these probabilistic methods to accurately describe the experimental data you're seeing. Then the other method that's used in Lattice Microbes is the reaction diffusion master equation. And so I have this diagram down here, which I think really outlines it when you think about it through the chemical master equation. Is so your first term here is the chemical master equation that's solved in a similar, or I shouldn't say solved, is sampled in the same way that it was done for the CME and the RDME. But the difference is you have these spatial subvolumes that allow for the chemical master equation to be solved in each one directly, assuming those environments are well stirred. Then your other term is a diffusion operator, which, similar to the chemical master equation in terms of sampling reaction states, is it samples spatial states described by the vector x. And so you have some sort of diffusion constant that allows transitions between your spatial regions. And so within the cell, and this is where I was talking about with the spatial regions, you can define different components of the cell to allow for transitions maybe into the membrane, out of the cell, into the cell, and so on. And so here are just kind of examples where you can have a diffusion event out of state X, or a diffusion event out of state X again to either side. There's not one actually a diffusion event back into X, but the same kind of thing happens where you have your term out of X and your term into X based on diffusion events. And so here's kind of some examples of the spatial ideas that you can do in lattice microbes. So what I'm showing here is the reconstruction of a yeast geometry using lattice microbes. And so this will also be a part of the tutorial where you're able to get a better understanding of how you can construct physical representations of your systems. And I don't believe in LM 2.3 currently, or what you'll use today. You're not going to be able to look at anything in VMD, but with later releases, there's actually a plugin that allows you to look at these simulations in VMD, which is how this was generated. As you can kind of tell, those are your van der Waals particles, which are representing the site types. It looks a little bit better in our current edition, where it's more of that Minecraft looking representation where you have the actual square spatial voxels. And so continuing on to just some more examples of where you can implement spatial data into the simulations, as Zan kind of touched upon it. What you have here is, I believe this is actin filaments from a cell, and you have your reconstructed cryo-EM version of them, and then you can generate an equivalent structure in lattice microbes. And so what this is very nice for is it allows you to take experimental data exactly how it's presented by the experimentalist, and being able to implement it directly into simulations. So an example is if you had, let's say, another example would be this epithelial cell, but let's say you had this epithelial cell, you could reconstruct the geometry inside of lattice microbes, and you know this is a state that the cell was found in, and you can use this to simulate trajectories of some biological process. An example, the big one, as Zan showed in her talk, would be with the DNA configurations and using the ribosome configurations to build the DNA configurations to do the whole cell simulations she showed. And so what we know, or what's very beneficial about doing things in that manner, is allows you to take a state of the cell that was experimentally captured. You're not having to do some sort of random sampling or random placement of particles. You can say with some certainty, as you know, that this is the spacing or the distribution of how the particles would be in the cell. And so that is a very quick and brief overview of what LM can do and how it somewhat goes about doing it. Again, you'll get more of this in the tutorials. I'm going to try and take less of time to go over the last section, which is just the AWS. So that's how we'll be running these today. What you basically need to know from this is that it's a set of cloud computing services that is going to allow us to be able to run the simulations where you don't actually have the program, have to have the program installed. And so they have a wide array of services that they provide. This is kind of just some more basic information about it. I believe there is a talk later in the week that will talk more about the idea of using the AWS, I think more with respect to using VMD and NAMD. But they offer a lot of different ways. And so what it does is you are allowed to create instances. So again, each person will have their instance assigned to them. And what you'll be doing is using Jupyter notebooks to do all of this. And so at this point, I'm going to stop with the slideshow and go over to the Jupyter notebook. And so I'm going to show you really quickly how to do this, and then I'll let everyone do it. And then we'll kind of get a little bit more into the actual tutorial component. So you're just going to put in the IP you were assigned directly into your browser. This Jupyter notebook page will pop up, and then you put in the password, which is just LM123. I will put that into the chat so it's clear. So again, just LM123, and that will allow everyone to be able to connect. And so then what you'll do is you'll open up to this page. And so there's some extra things that are in here. Don't worry about the doc section, that's documents. And so that can also be gotten from the website. It's just more guides in terms of how to use all of the programs. Where you're wanting to go is tutorials and then PyLM. But before I continue on with that, what I'm going to do is, I think I sent it around the IPs already, but allow everyone to kind of connect, give you a moment, because again, that was a lot of information I quickly delivered to all of you. So I'll give you all a moment to connect, and then I don't want to mess up your name. Saeed, I believe is how that's pronounced. I'm going to put an IP for you in the chat. Just give me one moment.
I think another person joined the chat a couple minutes into your talk, Troy. So if anyone else doesn't have, if anyone is missing an IP address, just let us know in the chat.
Okay, so yeah. So if you scroll up in the chat, there will be a document that might have assigned you an IP. It's not guaranteed. I spelled your name wrong. I'm very sorry. That's bad. Yeah, I'll show you the IPs again. Give me 1 second. Actually, I'll do this. No, let me. So yeah, there you should all be able to see your IPs, and then the people who didn't, I will send you your IPs directly. I spelled your name wrong. And so and then at some point, if you can give some sort of verbal or text communication in the chat that you were able to log on, I'll continue going with how to connect and kind of what the tutorials are exactly, a little bit more information about that. Okay, seems like everyone's connected. So again, real quick, the docs, it's just documents about the user guide and things like that, which can also be accessed from our group website under software, Lattice Microbes. And so there's also ways to install LM 2.3 here. I would recommend, unless you're like trying to do something immediately, I would recommend waiting until 2.4 comes out. The installation process for that is significantly easier and is a little bit more of a cleaner process than 2.3 is. And so if you go under tutorials, we're going to be more in this PyLM directory, but to quickly touch on, so there's the JLM, which will allow you to understand a little bit more about what JLM allows you to do. And what that is it stands for like Jupyter Lattice Microbes. So it allows for a little bit more of, it's an LM Python wrapper that's a little bit more built to be used in Jupyter notebooks. PyLM can also be, but JLM allows for a little bit more like widget features and some things like that. The other thing at the bottom here is Python. And so that's a Jupyter notebook. It'll take a split second to load. That has just Python information in it. So if you're not too familiar with Python coding, it could be something you might want to take a look at. It'll take a little bit to load. The other thing I want to point out, and I'll just give this a second. So when my computer's about to die. Okay. So this is the Python thing, just real quick. For the person in the chat who's saying they were having complications logging in, what you want to do is, I think it was Delera, I believe is how you say that. You're going to want to type this IP into a web browser. So I'll give you a second to copy it. And so all you're going to do is type that into a web browser. And then once you do that, a page will come up. I'm just going to copy this person's. That'll ask you for a password, and then the password is just LM123. And once you do that, you should be able to get to where it says docs and tutorials. I did just log into someone else's. Do not do that. The problem with that is you'll basically get in each other's way. That's why each person has their own instance. What I just did there won't cause any problems, but the classic do as you're told, not as I do kind of thing. The password is LM123. So LM123. I'll also put that in the chat to make it a little bit clearer. And so there are no attendees. Yeah. And if you're having any problems, you can message me or Maddie and we can help you along. But to continue a little bit more, so this is a Python tutorial. It has a lot of useful things. If you're trying to get more familiar with Python, I would take a look at it. Again, these instances will be running until later in the night. So you'll have access to this outside of today's, just the workshop period today. The other thing that you can do is you can actually download these. You can download them as notebooks themselves, which you would need to have Python installed to be able to run, but you can also download it as a PDF. And so that you can use as more of a reference guide, because as you look, if you go through this, it's pretty well documented. And then the other kind of little quick thing to know about the Jupyter notebooks, just in general, is you can use restart run all, and that will run the entire notebook. If you're going to save this, I would do that, because what it will then do is it'll run each of these cells individually, or run them all at once kind of thing. And so the cells can be run by hitting Shift Enter to run them, or it can be run by doing this run command up here, and that's to run individual cells. And then the one last little thing about Jupyter notebooks, and again, I know this is a lot, so if there's any questions you have, put them in the chat or just interrupt me. That honestly is somewhat easier. You're more than welcome to speak during this. Is if you ever think you messed up the notebook and you want to fix it, if you go to revert last checkpoint, that'll basically save it in a current state. And so what that'll allow you to do is kind of be very free in how you go about trying to play with the tutorials. And if anything were to go wrong, you can just essentially hit restart or hit that checkpoint, and it's like you never did anything wrong. It's gonna be pretty hard for you guys to mess anything up. So don't obviously go about trying to mess things up, but feel free to try things out and experiment. That is the whole point of the workshop. And so,
sorry, Terry, when you have a sec, could I get the IP for Katerina?
Yeah. Oh, that was actually hers. That's kind of funny. Put it in for everyone. Good. Okay. Let me. I'm going to resend this just because.
Yeah, we didn't get I don't think we have that document.
Yeah, I think I sent it too early. I think it's a classic. You don't see things until the person joins. So I sent it when I first got in here, which was essentially useless. So there's also that document to grab IPs if you need it. And so to kind of get started on the actual tutorials, took a little bit of your time. I apologize, but again, I'll keep these running. Is you want to go into the PyLM, and so your tutorials here are the TUT 1.1 to 3.2. And so each of them is kind of targeting a different idea. What I'll do is go into this one first, which is just looking at a basic bimolecular reaction and solving it with ODEs. So this is showing you how you can kind of do something with ODEs, and then the next one will be doable in Lattice Microbes itself. And so the example of the restart run all is what you're seeing now, is it will actually run everything. And to answer your question about how long this will be accessible for, it'll be up until probably like 10 o'clock tonight. 10 o'clock Central Time is probably when I'll turn them off. They can be left up a little bit longer, but it wouldn't go longer than the workshop. And so again, you can download these things to look at. The only problem is you would need to have Python installed for this tutorial, and for the more for the stochastic tutorials, you would need Lattice Microbes installed. So they're somewhat not beneficial to download, but that Python one, I actually recommend doing the restart, run the entire thing, and then downloading that to use as a guide, especially if you're trying to get more familiar with Python. It goes into pandas and some other like packages that are pretty important. So what I'm going to go do is go to this second tutorial, and then I'm going to be quiet for a little bit because I'm sure you guys are somewhat sick of hearing my voice. This is the actual LM tutorial. And so what you're seeing here, for those not super familiar with Python, what it's doing is you're importing some packages that are needed, setting some of your variables, and then you're defining a CME simulation. And so the way that's done is you define the simulation, then you need to define the species in the simulation, and then you write your reactions. And so the reactions are pretty simple to write. It's your reactants, then your products. And so if I wanted to add a new species to this, all I would need to do is add a species to the list, doing like comma, next in the alphabet would be D. And what I could do is have this D species occur when there's some sort of decay. And then additionally, you have to define the particle counts. I'm going to remove this. And then what the last step is you're defining your simulation parameters. So what your right interval will be, so every 30 microseconds, how long you want the simulation to be. If a time is not defined, it's in seconds. So this will be for 30 seconds. And time not being defined is like using these converters to essentially convert a number in seconds to microseconds or femtoseconds or something along those lines. Then what it does is it prints out your simulation so you can recheck it. You see what your right interval is, how long the simulation will be run for, your initial particle counts, and then you actually can run the simulation. And so it should take relatively quickly. You're only running a single replicate. And then it goes into post-processing. I'd recommend reading all of this stuff. I'm just going through it quickly so you can get an idea of it, and then I'll let you guys go through it. And so what you're seeing here is a trajectory of both particles A and C throughout the simulation. And so why is that interesting compared to the last one? Is you can tell it's a much smoother system because you're simulating it using an ODE, so it's not stochastic, it's not able to capture the fluctuations. And in this version it is. And so actually these two should be analogous. So if I were to go up to replicates and increase this replicates, you should get almost identical results between the two, as long as your kinetic rates are the same and your initial conditions. So with that, I'm going to let you guys kind of just go through them. As I said, there are a handful of tutorials. So these are your more simpler ones. This is looking at lack genetic switch I mentioned earlier. There's a little bit of information about how to debug in the program, and then some spatial simulations in these later tutorials. Some of the simulations might take a little bit longer to run, and so you can actually see there's some pre-downloaded files. And like, for example, for this simulation where you're running an RDME, that would take like 10, 12 hours, depending on how long you ran it for. So we have those simulations preloaded. And so you can just change which one you're loading into, look at how the different concentrations, that's what those numbers there, the different inducer concentrations, how they impact the kinetics of the simulation. And so with that, I'll let you guys just kind of go ahead and navigate. Feel free to manipulate the programs in any way, change the reaction schemes, change the kinetic rate, kind of get an idea of how it impacts the kinetics of the system. And there are also some little assignment type questions to give you some sort of idea or some challenges type things to do within the tutorials. If you have any questions, again, unmute your mic, just shout it out and or send something in the chat, and Maddie and I will do our best to help you and give you an answer. And so with that, I'll let you guys go ahead. I'm actually going to run to the bathroom briefly. So if you need anything, just message Maddie or ask, and then I will be right back.
Maddie, hello.
Hi.
I came in a bit late. I don't know where I can get the tutorials. I want to download as a PDF somehow.
Yes. So I will, we're using the Amazon web server. So I'll send you your IP address where you can get that.
Okay.
Let's see. Sorry, just give me a second.
Take your time.
Okay, so yeah, I just messaged you your IP. So if you just want to go and put that into a web browser, and then it should prompt you for a password, and I will also message that to you.
Thank you, Maddie. I got it. Thanks.
Okay, great. And then if you go to tutorials, I think there should be one just generally on Python if you're not too familiar. And then there's one on JLM, another folder, but we'll mostly be working in PyLM today. So if you just want to go ahead and start with 1.1 and work your way through.
Okay. Yeah, I got it. So, and I'm gonna go over it hopefully. I'm at at work, in LA, and trying to do some teaching and also listening to you guys. So it's kind of hard.
Yeah.
It's lunchtime, so I can talk a little bit. Thank you.
Yeah, no problem. Troy, so we used up another IP address. I'll just message you so no one else uses it.
Say that again. Sorry, Manny.
Oh, sorry. So I just gave out another IP address. So I just sent a message to you so no one else uses it.
Okay, no, that's fine.
So how's it going, guys? This is Zan.
So, all right. I mean, other people in the crowd, you guys are the judges if it's actually going all right. But everyone at this point, we went through a little brief presentation to just kind of go back over some of the main kind of points. And then at this point, everyone is now just working their way through the tutorials, showed them kind of the Jupyter notebooks,
Okay.
how to navigate them, use, and then because it's up here, if you recall, Marcelo had written that Python intro, also showed them that in terms of, I don't know how what everyone's background is in using Python. So this could also be a useful tool because obviously trying to do all of this without knowing anything about Python would also be slightly difficult.
Yes, although my favorite command is run all.
Yes, yeah, I did show them that.
And that pretty much takes care of it, as long as you don't change too many things in it, because you always forget to reinitialize a cell and things can go wrong. Okay, then yeah, it looks like you've got a number of participants, and I hope they do try it, give a little bit of feedback. And if they have questions, they can put it also into the chat. So I think I'm going to go off and work on the revising the manuscript. And how long do you think you'll be there? Is it supposed to be until 4 or 3:30? What do you think?
I think 3:30 is when the first break is, and then there's a question section after that goes, I think, until like 5 or something like that. So
you know, we might ask at 3:30 if there are really any questions. If not, then we can let them go, because I think, as I said, in the past of these workshops, the participants come with their own problem that they want to work on. Since this is, primary emphasis is on molecular problems, I can imagine there'll be a lot more questions on VMD and NAMD, doing the molecular dynamic simulations. But we'll check again at 3:30. So
yeah, and I told them I'll leave the instances running too, so they can use them outside of the time. I'll shut them down basically when I go home tonight.
all right, but it won't be passed though today. All right.
Yeah, no, they will be shut down tonight.
Okay.
Definitely. So again, for everyone in the audience, anything you want to do with them or be able to like capture, make sure you do so before. I would say basically try to do it before 6 o'clock, because at that point they will be shut down at some point. And once they're shut down, any progress you made will be deleted. So it won't even be like we could re-get it back if you did something. And then also you won't have the ability to access the instances anymore.
Right. Okay, well, thank you guys very much, and I'll see everybody a little later then. Okay. Bye-bye.
Have a good one, Zan.
Mhm.
Ugh. Okay, so I'm gonna see here. Just a quick note for those of you progressing through the simulations. Nothing should take more than like 10 minutes to run. If it's taking longer than that, either something is wrong, which could be a possibility, or what could be happening is you could be running a system that is evolving a lot of particles, thus taking more time to for the simulation trajectory to evolve. That's all with the like basic parameters. If you decide to run like 1,000 replicates of something, obviously that's going to take a little bit longer than running the like initially or default 10. There's nothing wrong with that. Just keep that in mind. But it is set up in such a way that all of these things can be, each tutorial individually should be able to be doable in no more than 20 minutes each. And usually it's much less than that. So just a little heads up on that. So I'm sharing my screen again. Just quickly at this point, you might have gotten down to the later tutorials where it is construction of the cells. I just wanted to show or talk about it a little bit. So what this is showing, and again, this is tutorial 2.4, 2.5. So what this is showing how you can build a spatial simulation. And so all of the steps are kind of, as we've talked about, the idea of creating your regions, creating the reactions, defining your diffusion events. But what I would more so wanted to just show, and so this is a feature of the JLM, is these plots down here. So when you look at it first, it's just a cube. It's just a green surface. But what this is actually allowing you to do is actually look at what the simulation looks like. So what you should be able to do is, as I am scrolling, you should be able to see, you're seeing different slices of the cell construction. And then what you're seeing in those little dots, if I can scroll down, would be the proteins or ribosomes, depending on what I have selected. And so why something like this obviously is useful is if those of you in the audience, if you're familiar with doing molecular dynamics, you actually get to use VMD and look at a kind of physical representation of your system. With a lot of the things with lattice microbes and just numerical statistical mechanical methods in general, you're really just looking at graphs. So it sometimes can be a little bit harder to determine if something is going wrong in your system. For example, if you're running something in molecular dynamics and you see the system explode, you know something's going wrong. So this is just a nice little tool or built-in functionality of lattice microbes and its implementation to be able to see actual spatial representations of your simulation. And so you can change the axes you're on. And so this is just another tool that I wanted to make sure I stressed to you guys during this period. Sorry again to interrupt, continue going. And again, if you have any questions at any point, go ahead and ask them or send something in the chat and we'll hopefully be able to give you a sufficient answer. So we're approaching what would be the coffee break period because it's about 3:30 Central Time. Feel free to keep working if you would like. When we reconvene at 3:45, I will discuss one last kind of little thing with LM, and then you're free to continue working on the tutorial, or you can stop, obviously, because that would be more of the tutorial / Q&A section of the tutorial session. So with that, enjoy your coffee break, and I'll hopefully hear back from some of you after. And then Mariana, question for you. At the end of this, you just will terminate it and it'll be fine, correct? It's not, I don't have to do anything else.
Yeah,
Okay, that's what I thought. And thank you again for sending all that information before. It's appreciated.
Yeah, so hi, Troy. Are people still on break, or I think, are they slowly finishing the exercises? What do you think? Or Maddie?
I think we're gonna come back together at 3:45. Troy said he was gonna go over one more thing and then take any questions.
Okay, all right.
Yeah. It was just a wrap up period, and then I was going to show the SGRS example to just give them another
Another-
system where LM was used to study a biological system, but not something as grandiose as the minimal cell, where it's a lot more information going in. This is just a little bit more of a simple gene regulatory network. And I was just going to use the web page in the Jupyter notebook to show them that.
Oh, that's great. That's another nice example of the Jupyter notebook. Beautiful. Okay, so you said back at 3:45 is when you guys are gonna.
Yeah.
Okay, sounds good. Then I'll just put it on mute and turn off the video so I don't have to rejoin. And I'll be back. All right.
Okay. Sounds good. Okay, we're a little past, it's basically like 3:50 now, so those of you who are still here, what I would just like to show you now, and again, you can continue on with the tutorials, is just some, as I mentioned to Zan, is basically another kind of application of LM. So in those tutorials, you saw some simplified versions of some applications that we've done, and a lot of them there are publications. Like in the Lattice state, it'll link you to a publication, and I can quickly show that if I open it. Is this, you can get to this publication, which is basically what all of this tutorial is based on. And so what I want to discuss now is some work that myself and another graduate student, Lattice group, did in collaboration with a group here in microbiology of Karen Vanderpool, and then a group of TJ Hott, John Hopkins. And so what this is looking at a regulatory network of a small RNA that regulates the sugar transport in E. coli, so the PTSG gene. And so what you're seeing here is a schematic of the regulatory network. So you have some sort of transcriptional activity regulation with the gene being able to turn on and off, some sort of transcription event to generate the SGRS. And then you have a chaperone protein that can bind, and then it binds to the PTSG regulator in green here. That's able to then stops translation from occurring. And so why I want to stress this is because it's a very good example of what can be done with LM. So what I'm going to here now is on this website. Sorry, I'm clicking around a lot. Here on this website, it goes into some information about the paper, along with a link to the paper itself, and then some downloadable files. Again, you would need Lattice Microbes installed to use this, but what you can do, or what I was able to do, is I can load up the notebook. And so why this is so important is with the super resolution imaging approaches that are used, a lot of this is done and describes biology typically on an average. So what you have here is the SGRS count across 20 minutes and the PTSG count across 20 minutes after the sugar shock is induced. But what's more interesting is with these techniques now, you can get actual distributions of the data. So what you're seeing here is each of these time points is representative of approximately, let's say, 100 cells were measured. And so there's a distribution of data. That average does not actually directly reflect all of the copy numbers that are found. And so what I'm showing here are distributions of the SGRS and PTSG count in experimental, through the experiments. And so what Lattice Microbes allows is typically using some ODEs, you would have to fit to the averages. But what we were able to do with this approach, and so this is just cells running the simulations, but what I'm getting down here is more to the comparison of the experiment and theory, is you can compare the averages, which again, this would be doable in an ODE. And that's actually how the work was originally done, which was published by Jingyi Fei while she was a postdoc in Dr. Ha's lab at John Hopkins, actually here, I believe it was. I don't think he had left yet. But what's more important and what is really beneficial about using the Lattice Microbes simulation suite, and more so using stochastic simulations in general, is we are actually able to fit and compare to the, not the averages, I'm sorry, the distributions of the species, the RNA species throughout the cell cycle. So rather than just fitting to averages, we could fit to the distribution and see how well our kinetic model parameters and scheme were able to capture both the SGRS counts, as I'm showing here, and then the PTSG messenger counts. And so again, I put this link into the link to this page here. in the chat, and so that'll give you more information about the paper. But why, again, I just want to stress this because it shows kind of the power of being able to simulate something stochastically with all of the new and improved techniques that allow you to quantify biology. Describing it on average behavior is, I would say, no longer sufficient, / you can describe it on a much better distribution or population level dynamics. And so that's what we've been able to do here in this model. And so this is, again, just an example of kind of the power of stochastic simulations and lattice microbes to be able to integrate theory and experiment, which I would say, especially doing stuff with computational work, being able to explain theory or explain experimental results and also being able to recapitulate them with kinetic models is a very important feature and ability of theory / experimental work or computational work. I'm sorry.
Yeah. Hi, this is Zan. I just want to reiterate one of the points that Troy nicely made is the whole field is moving towards, with the most recent Nobel Prize on super resolution imaging, you can go beyond the mean. You don't have to just record what the average behavior is. Those fluctuations that we talked about in the messenger distributions or in the messenger counts, which are reported as a distribution of the messengers, can be seen experimentally. And so as a computational theoretical biophysicist, you want to be able to explain, well, how do those how does that distribution of counts come about? And in doing that study, one thing that Troy left out, they were able to look at the data and also determine in the population how many of the cells had undergone multiple replications of the DNA. And I think that's going to become even more important because most of the time you cannot synchronize cells easily. So it would be great if you were able to, out of the data, infer how many had just maybe one copy of the DNA or 2 or 3 copies of it. And so in that paper, they also plotted out where is the gene for SGRS? Where is the gene for PTSG, right? So a lot of valuable insight came out of that particular paper. And I found it funny that the modeling of the mean got into Science. And then it's taking a while for those journals to catch up to the importance of being able to learn from the fluctuations as well. And as I said, that's only been possible since the super resolution imaging experiments have become more commonplace, like STORM and SM-FISH. But they're here and they're being developed at every university. It's a standard technique. So if you want to do modeling at a whole cell level, this is something that you're going to have to treat gene expression stochastically. But yeah, that was a nice example to bring in, Troy. Thanks a lot. I appreciate it.
And then again, if anyone you can continue on the tutorials. And then if you have any questions, I will sit in the, I'll be in the Zoom call until I think it ends. I don't know if there's a specific time, but if no one has any questions, probably around like 4:30-ish, I'll maybe sign off. And then again, you can continue to work on or play around with the tutorials until approximately 7 o'clock tonight. So again, 7 o'clock Central Time, and then I will have them shut down. But thank you for taking the time out to go through the tutorials. And then again, if you have any questions, send a message in the chat or feel free to speak up now. Mariano. Might not be there. Hello. I don't know if he's here.